The overall goal of this procedure is to label cells expressing a tetracysteine and fluorescently tagged protein with the dyes FlAsH or ReAsH and to quantitatively analyze the results. This is accomplished by first transfecting a plasmid carrying a fluorescent protein and the target protein containing a tetracysteine tag into the cell line of interest. Next, the expressed protein is labelled with ReAsH or FlAsH, and the non-specifically bound dye is removed by washing with BAL to prepare for microscopic imaging. Then, a confocal microscope is used to collect fluorescent images of the cells with the ReAsH or FlAsH and fluorescent protein channels. Finally, the images are analysed using ImageJ to quantify binding of ReAsH or FlAsH to the target protein. Ultimately, results can be obtained that show the extent of ReAsH or FlAsH binding to the target protein in cells through the correlation of ReAsH fluorescence with fluorescent protein fluorescence.
The main advantage of this technique over existing methods, like simply tagging the target protein with GFP, is that it can track a second property of the protein, such as conformational change, in addition to its localization. This method can help answer key questions in the protein structure and function field, such as oligomerization events, conformational changes, and ligand binding events that alter the ability of the tetracysteine tag to bind ReAsH or FlAsH. We first had the idea for this method when we wanted to assess how small submicroscopic oligomers of mutant Huntington formed in cells. Visual demonstration of this method is critical, as the data analysis steps are difficult to learn because they are technically complex.